hypothalamus, pituitary gland, pancreas, and also parathyroid gland. The first character of these hormones that their half-lives in the circulation are in order of minutes. They have very short half-lives. Why? Because they are circulating freely in the blood. They are usually circulating unbound and in free forms only, except for one protein hormone, which we call the insulin-like growth factor 1, because it has a binding protein, which is called insulin-like growth factor binding protein 1, and that's why it has a longer half-life. So we can understand that when a hormone is bound to a binding protein, it will be having a longer half-life. Unlike if it's circulating freely. If it's circulating freely, it will be having a short half-life. These protein hormones, we call them hydrophilic. They are water-soluble. That's why they can circulate freely in the blood, but on the other hand, they cannot cross the lipid layer of the cell membrane. They cannot cross the cell membrane. That's why the receptors should reside on the outer surface of the cell membrane, because they cannot cross to inside the cell, so the receptors must be found on the cell membrane. So, if that's the cell, and that's the receptor on the cell membrane, and that's the hormone, it will bind to the receptor. But we know that the cell center or the cell command center is the nucleus. Any function by the cell should come from the nucleus. So how will this hormone reach the nucleus to give the instructions and start, generating functions of that cell or giving orders to the cell? This hormone wants to give instructions to the nucleus to start function. So it cannot cross the cell membrane. So it needs help. It needs help from what we call a secondary messenger. A secondary messenger. This secondary messenger, we have different types of secondary messengers, and this will be explained by Dr. Mewish in biochemistry session, okay? But that's just a quick idea. We have, for example, the cyclic AMP. It's one of the secondary messengers. This cyclic AMP, for example, will be activated once the hormone is bound to its receptor, and then it will move to the nucleus and then transmit the instructions by the hormone to the nucleus, and the nucleus will start to give the orders to the cell to produce or to inhibit functions according to the instruction given by the hormone. Also, from the characters of the protein hormones, that they are stored in intracellular vesicles as prohormones. So they are pre-synthesized as prohormones, and once they are needed, these prohormones are activated immediately and released into the circulation in the active form of the hormones. So these are the main characters of the protein hormones. Am I clear so far, everyone? I can say simply, first of all, we have to know that all the steroid hormones come from the godfather of all steroid hormones, which is the cholesterol. So the cholesterol is the father of all steroid hormones. These steroid hormones are secreted from the adrenal cortex, the ovaries and the testes, or generally the gonads, and the kidneys. And here, these kind of steroid hormones are hydrophobic. They are not water soluble. So to circulate in the blood, they need to be bound to specific carrier proteins and albumin. That's why their half-lives are longer than the protein hormones. And as I said before, the concept is that once the hormone is bound to a plasma protein, it will be having a longer half-life. So the half-lives of steroid hormones are in the order of hours to days.